Okay, we're going to go ahead and move on in the Unit 2.2 notes. In this video, we're going to specifically look at topic 2, which is about passive transport. Passive transport is one, I guess you can call it a category of movement across the cell membrane. And so we'll take a look at what this means. Before we get started, just some basic review of some chemistry stuff. When we're talking about a solution. So a solution, just to remind you guys of something you might have forgotten from chemistry, a solution is when you have a chemical dissolved in another chemical. And oftentimes, and especially in this class, the chemical that's dissolving the other chemical is water. So there's a chemical dissolved in water. That's a solution. Now that substance that's dissolved in the water, we call that the solute. The solute is the chemical that's dissolved in the solution. And then there's something called a solvent. The solvent is the substance that's dissolving that chemical, which, like I said, the solvent is going to always be water, at least in this class. So water is the solvent. And then there's like a specific substance, sugar, salt, something dissolved in that. That's the solute. And that forms a solution. So that's what a solution is. Now, solutions can have different concentrations. There's solutions that have a huge amount of solute dissolved in it. And then there's solutions that have a lot less solute dissolved in that. And so we can measure the concentration of that solution by seeing how concentrated or how close together all those solute particles are in the solution. If there's a lot of them dissolved in a small amount of solution, they're going to be all really crowded and really next to each other. That's what we would call a high concentration when you have let's just pretend it's sugar. You have a bunch of sugar dissolved in a small amount of water. That's going to be a very high concentration. All the sugar molecules are right next to each other. Whereas a low concentration is where you have not a lot of solute dissolved in that given amount of water. So you could have a certain amount of water with very little sugar dissolved in it. And those sugar molecules would be really spaced out. And so that would be a low concentration. So think of concentration as not so much as the amount that's dissolved in the water, but how crowded the particles are. And so, I mean, you don't need a lot of, if I have a small amount of water, you don't need a lot of sugar to make it really concentrated, to make the sugar really crowded inside that solution. It can taste really sugary and really concentrated with a small amount of sugar. But if I had a big amount of water, a little amount of sugar is not going to make it that concentrated. So again, don't think of it as the amount of sugar, because I can have a huge amount of sugar, but if it's in a big container of water, it's not going to be very concentrated. And so think of concentration as the crowdedness of the solute. How crowded are those solute molecules in the water? If they're super crowded and super next to each other, that's because it's really concentrated. If they're more spaced out, that's less concentrated. And so there's something called a concentration gradient, which is a situation where one area has a much higher concentration than another area. We call that a concentration gradient. Now, particles, if a particle is moving, we could say that a particle is moving down its concentration, or we say that it's moving along its concentration gradient. When we say a particle is moving down or along its concentration gradient, what we mean is that that particle is going from an area of high concentration to an area of low concentration, like this picture is showing here. But then you can also have particles who might be moving against their concentration gradient or up their concentration gradient. That means they're going from an area that has a lower concentration towards an area that has a higher concentration. So just make sure you guys know what those terms mean when I say something's going down or along its concentration gradient, which means from high to low concentration. Or if I say something's going up or against its concentration, that means it's going from low to high, like this picture is showing you here. So in passive transport, now let's get into it. This is when a substance is moving across the cell membrane without the input of energy. So when you have a specific substance going into or out of the cell and it doesn't require an input of energy, meaning that it just happens all on its own, we would call that passive transport. And so the reason a substance would be able to move across the cell membrane without energy is because that would only be true in a situation where that movement of the molecules is getting the system closer to equilibrium, meaning that both sides are getting closer to the same concentration. So if these molecules are moving in a way that's getting both sides to be closer in their concentration amount, that's going to be something that's passive transport. That's going to be something the universe wants to happen. The universe wants there to not be a concentration gradient. The universe wants both sides to be at equilibrium, to have the same concentration. And this movement of a substance in a direction that gets closer to that, to achieving that goal, that the universe is going to allow that to happen free of charge, no energy required. The universe wants that to happen. And so this is going to require that there's a movement of substance down its concentration gradient, or an area from high to low. So like in this picture, these particles, if I can get a lot of these particles to move towards this side, that's going to help us achieve the goal of getting closer to equilibrium everywhere, where everywhere has the same concentration and there's no longer a concentration gradient. And so that would be an example of passive transport. And so there's three specific forms of passive transport, or examples of passive transports. There's diffusion, osmosis, and what we call facilitated transport, or sometimes facilitated diffusion. These three examples are all accomplishing what I just said. They're getting both sides closer to equilibrium, and it doesn't require energy. And so let's start with diffusion. Diffusion is when a substance, there's a net movement, an overall movement of molecules down their concentration gradient. So moving from an area that has a higher concentration to an area that has a lower concentration. And that is going to happen until until we reach what's called dynamic equilibrium, which is when everywhere has the same concentration. And so we call it dynamic equilibrium because once all of the areas have the same concentration, the molecules are still moving. They don't just stop moving. They're still moving, but now there's no overall movement in any given direction. And so an example, a very simple example down here is like if I put a dye, a crystal made of dye here in this fish tank, there's going to be a really high concentration of those dye molecules where this cube is. And what's going to happen through diffusion is those molecules, the dye particles, are going to want to start moving to all these areas that have a lower concentration until it gets to a point where this whole fishbowl now has the same concentration of dye everywhere. But once it gets to this point, which we call equilibrium, the particles are still moving. They don't just stop moving, but now they're moving in no overall direction. Whereas before there was a net movement, there was an overall movement of dye particles from here towards these other areas. Whereas now they're all just moving very randomly. And so here's an example with an actual cell membrane. So if I have a certain substance out here, let's just say this is sugar. It looks like sugar to me, like glucose or something. And on this side of the membrane, there's a really high concentration of sugar. And on this side of the membrane, there's a really low concentration of sugar. Well, through passive transport, what's going to happen is these molecules are going to want to move in a direction that's going to get both sides closer to the same concentration. And so that would involve, in this scenario, involve these molecules moving to this side of the membrane. So I'm going to see an overall movement of these molecules moving to this side and more of them moving to this side. And that's going to keep happening where they're moving from an area of high concentration to low concentration until the concentration on both sides is now roughly the same. So if you guys look, the crowdedness over here and the crowdedness over here of sugar is about the same. And so now both sides have the same concentration. But once they reach equilibrium, we call it dynamic equilibrium because it's not like the sugar is no longer moving. Like these sugar molecules don't just randomly stop moving. They're still moving across the cell membrane, but now they're moving at equal rates of movement. And so now there's the same amount of molecules going into the cell and the same amount of molecules going out of the cell. It's balanced now. And so from this point on, they are still moving in and out or across the cell membrane, but there's not going to be a change in concentration because there's the same amount moving in as the same amount moving out. Because the rate of movement, the speed of movement is the same going in both directions, these concentrations are now going to stay the same from this point on. But that's only going to start happening once the concentration is the same on both sides. Then the rates will equal the same. Whereas before that, you can see this large arrow. That means like overall, the rate going into the cell is way faster than the rate going out of the cell. And that's going to keep happening until you get to this equilibrium. So that's one example of facilitated transport. We are accomplishing what the universe wants. We are moving things in a direction that gets us to equilibrium, where it's the same concentration on both sides. This does not require energy. Osmosis is another good example. This is a lot like diffusion, but in this case, it's the diffusion of the water, not the solute. So now we're going to look at water moving across a membrane and not the solute specifically moving across the membrane. And so whenever we're looking at a net movement of water across a membrane, we call that osmosis. And so usually water is always able to diffuse across a membrane. Even in a real cell membrane, the phospholipid bilayer, water can go through the phospholipids, even though it's polar. Because water is so small, it is able to squeeze through the phospholipid bilayer, just not very fast on its own, but it always can leak through kind of slowly because it is polar. It does have a hard time. But regardless, in any situation, water can always get across the cell membrane if needed. And that's helpful because sometimes the solute, the stuff dissolved in the water, can't get across the membrane. And so when the solute can't get across the membrane, that means the solute can't diffuse. There's no diffusion of the solute. And so now water is going to have to take on the responsibility to fix this situation, to get the system closer to equilibrium. So if you guys look at this picture here, we have two sides of this beaker separated by a semi-permeable membrane that certain things can go across and other things cannot go across. These dots in this pink liquid represent the solute. So this is the solute. On this side, there's a higher concentration of solute. On this side, there's a lower concentration of solute. And let's say in this situation, the solute, these dots, they can't get across the membrane. So these guys can't move across the membrane. If they were able to move, they would want to diffuse to the left to get the concentrations to be the same, to go from a high concentration to a low concentration. But let's say they can't, they're stuck. All of these guys are stuck on this side, all of these guys are stuck on this side. Now what can happen instead then is we can have osmosis kick in. Osmosis is the movement of water, and water can almost always diffuse across a semi-permeable membrane. And so what's going to happen if the particles, the solute can't dissolve across or can't diffuse across a membrane, you're going to see a movement of water instead. And water is always going to move to the side. You're going to see a net movement of water towards the side that has a higher solute concentration. So if you look at this picture, what's happening is water from the left side is overall moving to the right side, which the right side is the side that had a higher concentration. Now, the reason this is going to happen is because by water moving to the right side, it's going to dilute the concentration on this side. So we're going to make it less concentrated by just having more water over here. So we're still going to have the same amount of solute, but now it's going to be dissolved in more water. So if you guys look, this is a really high concentration. They're all really crowded. But look what happens now when there's more water on this side. They can space out more. So we actually changed the concentration of that side, not by the solute moving or changing the amount of solute. The amount of solute's the same, but by taking that solute and allowing it to be dissolved in more water, we have caused the concentration to go down on this side. This side is now less concentrated. Even though it has the same number of dots as right here, they both have the same number of dots. You can count all the dots if you don't believe me. But because now there's more water for those dots to be dissolved in, this concentration is less than what it used to be. And that's what water is trying to accomplish here. It's trying to make this side less concentrated. And by water going away from the left side, so now you guys can see there's not as much water on the left side, that's going to make the left side more concentrated. So by water going away from this side, these particles are now going to be more crowded. So the concentration of these particles on the left side went up, while the concentration on the right side, where the water went down. And they actually reached a point, if you look here, where the concentration is basically now the same on both sides. The concentration on this side and the concentration on this side is basically the same now. If you guys look at the crowdedness, if I asked you which side is more crowded, well, now it's about the same. And so this is equilibrium. Both sides have reached the same concentration. And it was accomplished not by the solute diffusing, but by the water diffusing, which we call osmosis. This net movement of water across a cell membrane is osmosis. And this, you guys, requires no energy. This will happen all on its own. If you actually did set up a beaker like this, you would see this side, against gravity, start gaining more water to get this. The universe wants this to happen, and it's going to happen all on its own without an input of energy, because now the concentrations are the same. And so this gets tricky. You guys got to remember, for osmosis, the water is going to move to the side that has a higher solute concentration. The side that has a higher concentration of solute, like salt or sugar or whatever it is, that's the direction that water is going to go. Okay, which again is passive transport. This is accomplishing the goal of getting to equilibrium. Okay, just like this previous picture was accomplishing the goal of getting to equilibrium. But in this previous picture, we were talking about diffusion of the solute, movement of the solute from high concentration to low concentration to get the concentrations the same. Whereas here, we're looking at the movement of water to the side that has a higher concentration to dilute that side and make the other side more concentrated until they're the same concentration. Hopefully that makes sense. And so that's what they're showing you right here in this picture. And again, this is all dynamic, meaning like everything's still moving. So at first there was a larger movement of water to the right side and not as much water moving to the left side. The rate of water moving to the right was greater than the water moving to the left. So over time, you start to see this side gain water. And then you get to dynamic equilibrium where now there's no net movement of water, but there's still a movement of water. They're just moving at the same rate now. Now there's water going to this side, but there's the same amount of water going to this side over time. And so now everything's stable now, but there's still movement. We call that dynamic equilibrium. And then this is, so like I said, water has a hard time going across the phospholipid bilayer. It can. So like water molecules, because they're tiny, they can slip through these phospholipids, even though the phospholipids don't like water, like the middle of the phospholipid bilayer. It doesn't like water, but water can slip through. It just happens really slowly. And so a lot of cells have evolved to have these special proteins, these special transport proteins called aquaporins. Aquaporins are these proteins that allow water to go into and out of the cell way more easily. And so that's what this guy is here. It's basically a channel for water to quickly move across the cell membrane so that this helps with osmosis happening a lot more easily inside the cell. Cells that don't have aquaporins, there's some cells who don't have aquaporins, osmosis is going to happen a lot more slowly because water is going to have a harder time going through these phospholipids. And so there's three situations that a cell could be in terms of osmosis. And in terms of the type of solution that it's in. And so cells can be in a solution that's isotonic, hypotonic, or hypertonic. And so this hopefully sounds familiar from freshman bio. Isotonic is when a cell is in a solution that has the same concentration as inside the cell. And so the concentration inside and outside the cell is basically the same. So there's no net movement of water. There's water going into the cell, but there's water going out of the cell, and that's happening at the same rate. And so that's what's going to happen in an isotonic solution. Now in a hypotonic solution, the prefix hypo means below or less than. Just a good prefix to know for science words. Hypo means less than or below. And tonic means concentration. And so hypotonic means less concentrated. So when I say a hypotonic solution, I'm talking about a solution that's less concentrated, hypotonic. And so cells can sometimes find themselves in a hypotonic solution where it's less, the solute concentration is less on the outside, and there's a higher solute concentration inside the cell, as represented by these purple dots. And so since the inside of the cell is more concentrated, water is going to want to move to where it's more concentrated, right? Because water is going to try to dilute the side that's more concentrated. So in this case, there's going to be a greater movement of water into the cell than out of the cell. This is going to cause the cell to expand and gain water. And that's going to be good and or bad depending on the cell. So like animal cells, this is not good for animal cells. Animal cells are going to explode probably if they're in a hypotonic solution, because as they gain water, they're going to gain so much water that they're going to rupture, or the term is lysing. The cell's going to lyse and break open. And so the cell's going to die if it's in a hypotonic solution. But actually for plant cells love hypotonic solutions because plant cells won't explode. And that's because they have a cell wall, around their cell membrane. So if you remember, plant cells have a cell wall. That cell wall is very rigid and strong. And so water is going to go into the cell, but it's not going to be able to explode the cell or lyse the cell because of that cell wall creating a lot of pressure on the inside. And I'll show you a picture of that in a second of what that looks like. But we say that that causes turgor pressure in plant cells, which makes the plant cells really pressurized, which plants like that because it helps them to like grow upward. Like if you guys ever put a flower in a vase, when the plant cells are all turgid, you have this nice crisp stem holding up the flower. But if there's not a lot of pressure in those cells, then the flower is going to start to wilt and then the cells aren't pressured enough to keep it upright. Because plants don't have like a skeleton system, for example. So that's a hypotonic solution. And then a hypertonic solution is the opposite. So the prefix hyper means more than or greater than. So think of like a hyperactive kid or someone who's very hyper or has a lot of energy, more energy than a normal person. That's what hyper means. And so in this case, we're looking at a solution that has a higher concentration, hypertonic, higher concentration. And so in this case, the cell is in a solution that's more concentrated than inside the cell. So inside the cell, it's less concentrated. Outside the cell, there's a higher concentration. And so water is going to want to go out of the cell to where there's a higher concentration, because water will always want to go where there's a higher concentration of solute to try to make that dilute. And then, so in this case, you'll see water is going in, but there's a lot more water going out of the cell. So overall, the cell is going to start shrinking and shriveling up. And this is not ideal for anyone. Cells do not want this. They don't want to be in a hypertonic solution where they're all shriveled up like this, because this usually makes them start to not function properly, and then they end up dying. And so here's a picture that kind of summarizes this for plants and animal cells. So in an isotonic solution, this is actually great for animal cells like isotonic solutions, where they're not gaining or losing water overall. It's just a nice balance for them. Whereas plant cells, they would prefer a hypotonic solution because they're going to fill with water, but that cell wall is going to help pressurize the cell and prevent it from exploding. So we say that the plant cell is turgid. Plant cells like that. But in animal cells, if they fill up with too much water, they're going to lyse or explode. And then in a hypertonic solution, you can see that animal cells are going to shrivel up because they're going to lose water because it's more concentrated outside in the solution. The solution is hypertonic, more concentrated. And in a plant cell, it's also going to shrivel up. We call it plasmolysing in a plant cell. Plasmolysing is when the cell membrane pinches inward from the cell wall. So you have the cell wall and then you have this pinching inward of the plasma membrane. We call that plasmolysed. But it's the same situation where that's not good. Like plant cells don't want that, animal cells don't want that. And so make sure you guys memorize these words and you know what that hypotonic means less concentrated and hypertonic means more concentrated. And be careful with how the word is used. So a lot of times these words are used to describe the solution that the cell is in, but you can use these words in different ways. So for example, in this picture, this is what confuses students sometimes is they don't know what the word means, but they memorize like a specific scenario with the word. So for example, like in this beaker, I would say that the solution that the cell is in is hypotonic because it's less concentrated. But then I could say that the cell is hypertonic. So the cell is hypertonic to the solution. It's more concentrated than the solution. And the solution is hypotonic. So we would say in this picture, the solution is hypotonic, it's less concentrated, and the cell is hypertonic. And so make sure you understand the word. Now, usually when we're using these words, we're describing the solution, but they might be describing the cell. Like if I say the cell is hypertonic, I'm not talking about this. I'm actually talking about this picture where the cell is more concentrated, and then this solution is hypotonic to the cell. So just be careful when you're reading certain problems or scenarios using these words hypotonic, hypertonic, or isotonic. And then the last thing on here is facilitated transport. Facilitated transport is when you have a movement of a solute across a cell membrane from high to low. from a high concentrated side to a lower concentrated side, which is what the universe wants, because that will allow both sides to get closer to the same concentration. But this is when you need the help of a protein. And so whenever a protein is needed to help with diffusion, we call that facilitated diffusion or facilitated transport. There's a facilitator. The facilitator is the protein, either a channel protein or a carrier protein who's helping move those things across to the other side. And so whenever you have a solute like sugar or whatever it is moving across the cell membrane from an area of higher concentrated from a higher concentrated side to the lower concentrated side, but it's using the help of a protein, we call that facilitated transport, which also doesn't require energy. So these proteins are helping these molecules move in a direction that the universe is happy with. And it doesn't require energy for these proteins to help these guys move. Like they'll just naturally assist in the movement of these guys as long as they're moving from high to low concentration to get the sides closer to equilibrium. So it's basically diffusion, but with a helper, okay? Now, when there is no helper, like this picture way back here, we just have a movement directly across the cell membrane without a protein. We would just call that simple diffusion. So this is just simple diffusion. They're just simply moving across the cell membrane from high to low until you get to equilibrium. Whereas if there's a helper, which a lot of molecules do need a helper, they need a facilitator or a protein, that would be facilitated transport or diffusion. And then it talks about the difference between a channel protein and a carrier protein. Channel proteins have these open channels, and so these guys move very quickly. And so if there's a solute can just very quickly flow in and out of the cell. But a carrier protein, remember the difference, a carrier protein actually has to bind to the solute and move it to the other side. So these guys are a lot slower usually. And so if they have to actually bind to this guy and then be moved to the other side, they can usually only go one at a time. So one of them has to move, then the next one can go, then the next one can go. Whereas in a channel protein, it's a lot quicker because they can just go one after the other, or like 5 at a time or 10. Like they can just zoom through the channel protein very easily. Not so easily with the carrier protein. But regardless, as long as it's a movement of molecules from high to low, that's going to be passive transport, no energy required. And so this picture summarizes all of that. So here's simple diffusion going from high to low, which is good. The universe wants that until both sides get to equilibrium. Here's facilitated transport with a channel protein or a carrier protein helping to move substances from the more concentrated side to the less concentrated side, which is good. That's what the universe wants. And here's osmosis, which is the movement of water across a cell membrane. All of this involves substances moving down their concentration gradient, getting closer to equilibrium. No energy required for any of this. So that's it for these notes. Hopefully you guys can think through all these different scenarios on these questions. And then we'll talk a little bit about this in class because it does get tricky. So that's it for now. Thank you guys. See you later.